Welcome back to the Deep Dive. We are jumping straight in today, folks, because the topic is, well, it's urgent. Global plastic pollution. I mean, the numbers alone are just staggering. Think about this. Over 400 million tons produced every single year. And the amount that gets properly, effectively recycled, it's less than 10%. It's tiny.
It really is. It's almost negligible when you consider the scale.
So you've got this mountain, this colossal amount of plastic sticking around in ecosystems for, what, hundreds of years?
Yeah.
The old ways, melting, sorting, the usual recycling, they're just not cutting it.
No, they're overwhelmed. And that's why we need something genuinely different, a fundamental shift. So today, that's what we're diving into. Plastic-eating enzymes.
Okay.
Now, these aren't science fiction. They're real biotechnological tools. Basically, proteins, sometimes natural, often engineered now, that can actually break down these incredibly tough plastic polymers. Eco-friendly, too.
Right. So our mission for this deep dive is to really unpack that.
Yeah.
How do these things, these biological catalysts actually work? And where do they even come from? I heard it's kind of an accidental discovery.
We'll get into that amazing story, yes. From a waste plant to, AI-driven labs.
And crucially, what's stopping them? What are the hurdles between this amazing tech and actually solving this global mess?
Exactly. The potential versus the reality.
So yeah, get ready for a deep dive into some fascinating biochemistry that could genuinely change how we deal with waste.
Okay, so let's break down the how. To really get the potential, you need to understand the core problem. Plastic polymers, right? These super long chains of molecules.
Very stable. Very hard to break apart naturally.
Precisely. So for waste, you need something that can, snip those chains effectively. An enzyme is basically nature's catalyst. It speeds up a very specific reaction. In this case, its job is degrading these synthetic plastics.
Right.
It targets the chemical bonds holding the chain together.
And breaks them.
Turns them back into the original building blocks, these small molecules called monomers.
Okay, monomers.
Uh-huh.
And you mentioned the mechanism. A lot of the buzz seems to be around P-tape plastic. That's polyethylene terephthalate.
That's the one.
Your water bottles, polyester clothes. Why the focus on PT? And how does the enzyme, physically attack it?
Good questions. PT is, relatively speaking, one of the easier plastics for enzymes to digest. Plus, it's everywhere, so it's economically significant. The enzyme itself, it physically attaches to the surface of the polymer. Think of it like a tiny molecular machine finding the right spot.
Like a key in a lock.
Sort of, yeah.
Yeah.
A very specific fit. Once it's attached, it kickstarts a reaction called hydrolysis.
Breaking with water.
Exactly. It uses water molecules to break those specific chemical bonds in the long PT chain. Snip, snip, snip.
Okay, and this is where it really differs from just melting down plastic, right? Melting damages the material. What do you get at the end of this process?
This is the crucial part. You get two specific small molecules. Terephthalic acid, TPA, and ethylene glycol.
TPA and ethylene glycol.
And these are the original monomers used to make the PET in the first place. This is the win for a true circular economy.
How so?
Because when you melt plastic, the polymer chains get shorter, weaker, you downcycle it, a bottle becomes carpet fiber maybe, can't easily become a bottle again.
Right. Lower quality.
But with these pristine monomers, you can purify them and feed them right back into the process to make brand new, virgin quality PET plastic.
So bottle-to-bottle recycling properly.
That's the dream, the closed loop that everyone's been chasing.
Okay, that does sound almost too good to be true. If it was that straightforward, you'd think recycled plastic prices wouldn't be all over the place. Let's rewind a bit. Where did this idea even start? You mentioned an interesting discovery story.
Oh, it's a great story. Happened back in 2016. A team of researchers in Japan, they were literally looking at samples from a plastic bottle recycling plant.
Just digging around the muck.
Pretty much. And they found this bacterium living right there in this peat-rich environment that had naturally evolved the ability to eat peat plastic.
Wow. Evolution in action.
Exactly. They named it Ideonella sakaiensis. It's amazing, isn't it? One of the biggest potential breakthroughs for this environmental nightmare came from, essentially, industrial compost, not some billion-dollar lab initially.
So, Ideonella sakaiensis. And the key was the enzyme it was producing.
That's right. The star player was an enzyme they called PTase.
PTase. And what made that specific enzyme special when they first found it?
Well, just finding it was huge. But technically, the cool thing was its ability to work at pretty normal ambient temperature.
So no need for tons of heat.
Right. which avoids massive energy costs associated with other chemical recycling methods. But, science is rarely simple. They quickly found out PTase worked better with a partner.
The sidekick enzyme.
Kind of, yeah. Further research showed that another enzyme produced by the same bacterium, called M-hetase, worked together with ketase, a synergistic effect. Meaning, when you put both PTase and M-hetase together, they broke down the peat much faster and more completely than PTase could on its own. It's like a specialized demolition crew.
Okay, so we find one key, PTase, but realize we need a whole set of tools, like M-hetase. And we have to remember, PET is just one plastic type, albeit a common one. What about all the other plastics out there? The really tough ones.
That's the next big challenge, exactly. Peat is like the low-hanging fruit in some ways, but the wider plastic world, much tougher. Scientists are looking at other enzymes too, things like cutinase for different types of polyesters, and lacase and peroxidase. They're exploring those for harder plastics like polyethylene PE.
PE, that's our plastic bags, milk jugs.
Yep. And polypropylene or PP yogurt cups, car parts, and these are chemically much more resistant, much harder to break down.
So it's not a one-size-fits-all enzyme solution.
Not at all. You need a different enzymatic key for almost every different polymer lock. That's why so much research now is focused on engineering and tailoring these enzymes.
Right. And you mentioned that shift from finding it in nature to actively engineering it in the lab.
Right.
Using things like AI-driven protein design. What does that actually involve for someone not familiar with that?
Okay, so once they had the natural PTase sequence, the goal became, how do we make it better, faster, more stable? They use techniques like high-throughput screening. Basically, they take the enzyme's genetic code, make thousands of tiny changes, mutations very rapidly.
Trying lots of variations.
Exactly. And testing them all simultaneously to see which ones work best under certain conditions, maybe higher temperature or faster speed. Now, couple that rapid testing with machine learning, with AI. The AI can analyze the results, spot patterns, and actually predict which future mutations are most likely to improve the enzyme even more.
So it massively speeds up the optimization process.
Dramatically. Instead of just trial and error, it's guided evolution, essentially, creating hyper-efficient variants much faster than nature ever could.
Which brings us neatly to the advantages. Why is this tech so exciting? We know it yields those pure monomers for true recycling. What else? What's the big sell beyond just being green?
I'd say the biggest practical advantages are probably energy savings and the lack of toxic chemicals.
Okay.
Traditional chemical recycling often needs nasty solvents or really high pressures and temperatures. Here, we're mostly talking about using water under mild conditions, often near room temperature, especially with those initial PTase discoveries.
That's a huge energy saving right there.
Massive. It's fundamentally more energy efficient.
And does the enzyme's specificity help with the contamination problem that plagues normal recycling? When different plastics get mixed?
Absolutely. That's another key benefit. Because the enzyme is designed to target only the specific bonds in PT, for example, it tends to ignore other contaminants or different plastic types.
So you get a cleaner end product.
Much cleaner. The resulting monomer stream, the TPA and ethylene glycol, is incredibly pure. And that purity is what allows you to make that high-quality, virgin-grade plastic again. It makes the whole process economically much more viable.
Okay, fantastic. Sounds brilliant. Green, energy efficient, circular. Now, the reality check. What's holding this back from being everywhere? Why aren't all our plastic bottles being munched by enzymes right now? What are the roadblocks?
Yeah, there are definitely significant hurdles still. These are the critical engineering and scaling challenges. The number one issue historically has been the rate of degradation.
Speed.
Natural enzymes, even the first PTase, were relatively slow. We're talking days, maybe even weeks, to break down a meaningful amount of plastic.
And industrial recycling plants need to process tons per hour.
Exactly. If your process takes weeks, the economics just don't work for the sheer volume of waste we generate. That's been a major focus for improvement.
Okay, speed is one. What else?
Stability is another big one. Enzymes or proteins, remember, they can be fragile.
They can break down.
They can denature, yeah. Unfold and stop working, especially under harsh industrial conditions, fluctuating temperatures, different pH levels, chemical residues in the waste stream.
So you need tough enzymes.
You need robust enzymes. And then there's cost. Producing huge quantities of highly purified enzymes reliably and cheaply. That's a major biochemical engineering challenge in itself.
Right. And you touched on this earlier. The fact that most enzymes developed so far are great for PE, but what about the really common ones like polyethylene, PE? and polypropylene key, they're much tougher nuts to crack, enzymatically speaking.
They absolutely are. They lack the specific chemical bonds, like the ester bonds in PT, that current enzymes like PTase target easily. That chemical inertness makes them far more resistant. So finding or engineering enzymes for PE and PP is a massive ongoing effort. It's a huge bottleneck.
And finally, what about dirty plastic? Does the enzyme care if the bottle still has soda residue in it?
It can matter a lot. Even small amounts of contaminants, food waste, labels, other plastics mixed in can significantly inhibit the enzyme's activity. It reduces efficiency. So, cost. All these practical issues slow down widespread adoption.
Okay, but this is where the really exciting stuff is happening now, right?
Yeah.
The innovation pipeline. Using that AI and synthetic biology to tackle these exact problems, speed, stability, selectivity, how are they doing it?
This is where the pace is just incredible. They are literally re-engineering these molecules using techniques like directed evolution, which is like hyperspeed natural selection in the lab.
Constantly refining.
Constantly refining the enzyme structure and combining that with machine learning predictions. For speed, a lot of work focuses on tweaking the enzyme's active site.
The part that actually binds to the plastic.
Exactly. They redesign that little pocket so it grabs onto the plastic polymer chain much more tightly, or positions it perfectly for the chemical reaction to happen faster. We're talking improvements that make the reaction happen hundreds, even thousands of times faster.
Going from weeks to break down plastic down to hours.
Potentially, yes. Some of these engineered superenzymes can chew through PT in a matter of hours now, just starting to look industrially viable.
Wow. Okay, that tackles speed. What about stability? Making them tougher and reusable.
Right, for industrial scale, you need enzymes that can last and be reused. Two key strategies here. One is creating thermostable enzymes,
Heat resistant.
designed to work effectively at higher temperatures. This is actually clever because a bit of heat helps loosen up the plastic polymer chains, makes them more flexible, easier for the enzyme to access and break down.
Sort of pre-softening the plastic.
Kind of, yeah. And the other major innovation is enzyme immobilization.
Immobilization.
Yeah.
Sticking them onto something.
Pretty much. You chemically attach the enzymes onto a solid support material like tiny beads or a membrane. This protects the enzyme, makes it more stable, and crucially allows you to easily keep the enzymes in the reactor and separate them from the liquid product stream so you can reuse them over and over.
Making the whole system more efficient and cost-effective.
Exactly. Both thermostability and immobilization are key for making this work at scale.
And this isn't just theory anymore, is it? We're seeing companies actually building facilities based on this.
Absolutely. It's moving out of the lab. You've got companies like Carbios in France, for example. They're building commercial-scale plants using enzymes to recycle PA. It's proof that the technology and the economics are starting to align, at least for PA.
And the impact goes beyond just plastic bottles, right? You mentioned textiles.
Yes, huge potential there.
Yeah.
A lot of clothing, particularly sportswear and fast fashion, is made from polyester, which is PE.
Oh.
So enzymatic recycling offers a way to take old polyester clothes and break them right back down to those pure monomers, TPA and ethylene glycol, to make new, high-quality polyester fibers. Real textile-to-textile recycling.
That could transform the sustainability of the fashion industry.
Potentially, yes. And thinking even bigger, beyond just contained recycling plants. What about the plastic already out there? The pollution in oceans, landfills, those pervasive microplastics.
Can enzymes help clean up that existing mess?
That's the hope with bioremediation. It's a bit further out, perhaps, but the idea is to use either the enzymes themselves, maybe in spray form, or even engineered microbes designed to produce these enzymes to target and break down plastic pollution directly in the environment.
Tackling the legacy pollution.
Exactly. Cleaning up the microplastics that have escaped our collection systems. It's a critical area if we want to actually heal damaged ecosystems, not just manage future waste better.
So we've got the initial discovery, the clear advantages, the challenges being tackled by some seriously advanced engineering. Where does this head next? What's the ultimate vision when you combine this with cutting-edge biotech like synthetic biology?
The long-term vision gets pretty advanced. We're talking about using synthetic biology to essentially design bespoke microbes. Bacteria that are engineered to be highly specialized, self-contained little biofactories for degrading plastic.
Not just producing the enzyme, but being the whole system.
Potentially, yes. Using tools like CRISPR gene editing, scientists are exploring ways to engineer bacteria to not only produce the necessary enzymes efficiently, right where they're needed, but maybe even to consume the resulting monomers, the TTZ and ethylene glycol, as their own food source, turning the waste plastic directly into bacterial biomass or other useful compounds.
Okay, wow. But hold on. Engineering bacteria. Safety, unintended consequences.
It absolutely does, and it should. The ethical considerations and safety protocols here are paramount. Biocontainment isn't an afterthought. It has to be baked in from the start.
You can't risk these engineered organisms getting out and messing with natural ecosystem.
No way.
Yeah.
The potential for using them is huge, say, in a contained landfill bioreactor, but the risk of escape needs serious mitigation. We're talking about robust safeguards.
Like what? What kind of concrete strategies can ensure they stay where they're supposed to?
Well, things like building in genetic kill switches. These are genetic circuits designed so the engineered microbe can only survive under very specific conditions found inside the processing facility, maybe requiring a synthetic nutrient or a specific temperature. If it gets out, it dies.
Okay. Built-in self-destruct.
Essentially. And alongside that, rigorous monitoring is crucial. We need absolute certainty that all the byproducts generated throughout the entire process, even from these super efficient new enzymes, are completely non-toxic and safe. No trading one problem for another.
That feels like the central tension here. The science, especially with AI helping design these proteins, is moving at lightning speed. Regulation, public understanding. That often lags way behind. So here's a final thought for you, the listener, to chew on. Given that AI can now potentially design these hyper-efficient plastic-eating enzymes faster than ever, maybe in months, what steps do we need to prioritize right now? What regulatory frameworks, what public education, what safety checks need to be put in place urgently so we're actually ready to deploy these potentially revolutionary tools safely and effectively on a massive scale before the plastic problem gets even worse? Something to think about as these super enzymes get closer and closer to changing our world.